In January 2024, Chinese scientists reported that they had successfully cloned a research monkey in 2020, and now that monkey had reached adulthood at almost 2.5 years of age. So how did scientists succeed in cloning this monkey? In this video, we are going to take a look at the science behind cloning. Specifically, we will examine what happens in normal development. How do you clone with the process of artificial twinning? And finally, how do you clone with nuclear transfer? In order to understand the process of cloning, it's first necessary to take a look at the normal process of development. Development begins with fertilization, when sperm cells come into contact with the egg and basically join the sperm cell nucleus with the egg cell nucleus. In doing so, that combines genetic material, or genes or DNA, from the mother with genes and DNA from the father. This results in a fertilized egg, which has a unique combination of genes which will direct unique characteristics for that individual. Now, with this single fertilized egg, ultimately, it's going to develop into the new life form. But what's going to happen along the way is that original cell is going to undergo cell division in a process scientifically called mitosis. And so we're going to go from a single cell to 2 cells to 4 cells. That process is going to continue until we end up with a ball of cells over here. Now, what's going to happen with normal development is that this ball of cells is going to rearrange itself. It is going to implant into the wall of the uterus, and we're going to go through a process of embryonic development where the embryo is going to basically create a primitive form of all of the adult organs. And then once the embryo stage is over, it is going to go into a fetal stage where those preliminary organs are going to go through additional development. Ultimately, the fetus is going to be born, and the process of labor and childbirth to deliver a hopefully healthy baby. Now, what is important to recognize is that all of the cells in this embryo, all the cells in the new organism, are going to have the same genetic material. They're going to have the information that's coming from the mother and coming from the father. But depending upon what those cells are going to become, the DNA is going to direct their differentiation or development. So muscle cells are going to develop muscle characteristics because of the muscle genes that are present are turned on. Skin cells are going to develop skin characteristics because the genes or the DNA for building skin type structures is going to be directing the differentiation of that cell, the development of that cell. One of the earliest forms of cloning was a form called artificial twinning. And so again, in order to understand what occurs in the process of artificial twinning, we need to look at what happens with the creation of an identical or monozygotic twin. So if we take a look at this, again, focusing in on the early stages of development, we've got the sperm cell coming in, joining with the egg, and so the nucleus with the DNA, the genetic material from the mother, is going to be combined with the nuclei of the sperm, with the DNA and the genetic information from the father, resulting in this fertilized egg here with this unique combination of genes that are going to be present, a combination of genes coming from the mother and coming from the father. Now again, during normal development, all of the cells of the adult body are going to develop from this single fertilized egg. What happens in the process of monozygotic twins is that this egg, as it is going through the process of division, it is going to result in a splitting of this ball of cells, in essence, resulting in kind of 2 clusters of cells that are going to be present. Each one of these clusters of cells, again, has the same genetic material, the same information in the nucleus as that original cell. So to summarize the previous slide with the information on normal development, we go from that fertilized egg through a series of cell divisions to 2 cells to 4 cells, ultimately producing a ball of cells over here. And this ball of cells is going to continue to develop into the embryo, producing the primitive organs, and then through further development, ultimately produce the offspring over here. Now, in the process of artificial twinning, what's going to happen is that this ball of cells, in essence, is going to be split in half. And so we're going to have one cluster of cells kind of over here, and another cluster of cells up there. And so each one of these clusters of cells is then going to produce an identical twin. These are going to be monozygotic twins because they're coming from a single fertilized egg. So in the process of artificial twinning, we are going in, scientists are going in, and then splitting the embryo at a very early stage. And because of that, that is going to result in the two different individuals being produced. And again, these are going to be identical twins or monozygotic twins. Now, the process of artificial twinning has been used for cloning for many, many years, basically about 140 years now at this point. It started out in the 1880s when a scientist was able to separate the cells of a sea urchin embryo and result in two separate genetically identical sea urchins being formed. This continued in the 1930s and 40s when scientists were basically splitting the embryos of amphibians like frogs and salamanders. And then more recently, this has been used to clone organisms like monkeys in the early 2000s. Now, the process of artificial twinning works great if you're working with a fertilized egg or potentially working with very, very early stages of embryos. Because basically you're starting out where these cells are in the process of dividing to produce all of the cells in the new organism, the new body that's being produced. Again, if we split that early on, the body basically kind of compensates for it. It basically replaces those lost cells and results in a full-size organism being produced, in this case with the same genetic material, so they're clones with one another. But what happens if we want to clone an existing organism, not an organism that's being produced through the process of fertilization? In that case, we need a different method. If we're going to clone an existing organism, what we have to do is transfer that genetic material, basically the material in the nucleus of their cells, into another structure in order for that structure to develop into a new embryo that will develop into that cloned organism. And so in the process of nuclear transfer, what happens is we start out with an oocyte that's been donated, and so much like in vitro fertilization. But once we get that oocyte out in the laboratory, we're going to remove the nucleus. We are going to remove the genetic material from that egg cell. Now, under normal circumstances, the information in the nucleus of the egg cell would combine with the information in the nucleus in the sperm cell, resulting in kind of that new kind of genetic combination for that organism being produced down here. In this case, though, we're not going through the process of fertilization, so we're going to eliminate the egg nucleus, and instead, we're going to replace it with a nucleus of an existing organism cell. And so in this case, we're taking fibroblasts, which are skin cells, we're growing them in the laboratory, and we're going to collect a nucleus, again, the nucleus containing the genetic material, and we're going to take that completely and transplant that into that enucleated egg, into the egg cell that doesn't have a nucleus in it. In that way, that nucleus that's been transferred into it, nuclear transfer, is going to direct the development and differentiation of the cells that are ultimately going to produce the infant over here, this cloned infant, coming up with the same genetic material as the original donor cells up here. So in the process of nuclear transfer, we start out with an egg donor over here, and the egg donor is going to produce an egg that's represented in here with blue, so we can see the blue cell, which surrounds a blue nucleus over here. We're going to have the nuclei donor over here in red, and this is basically going to be the organism that's going to be cloned. And so we've got a red cell here with a red nucleus. What we're going to do in the process of nuclear transfer is that we're going to go through and we are going to eliminate the nucleus out of our egg cell, and we're going to take the red nucleus, the nucleus of the donor cell, this organism that's going to be cloned, and we're going to combine that into a situation where we've got the egg here, again represented in blue, with the red nucleus coming from the donor over here, the organism that's being cloned. This red nucleus then, with the genetic information coming from the donor, is then going to go through a series of cell divisions, all of which are going to result in cells with these red nuclei. The red nuclei then are going to direct the development of the organism, and so we're going to end up with a cloned copy of the individual over here. And so this is a genetically identical organism to the original donor over here, the nuclei donor over there. So now with this better understanding of the cloning process, we can consider, is cloning something that we need to do? There are claims that cloning monkeys and other organisms will accelerate biomedical research, but are there ethical considerations that we need to think about prior to advancing the use of cloned animals? Thank you for watching this video on the science of cloning. The video to the right goes into some of the ethical questions involved with the 2024 announcement that Chinese scientists had cloned a research monkey. Thank you and have a great day.